Yeah, thanks, Max and Uri. When I realized I was coming to Copenhagen for this conference, and I saw that there was a physics of aging workshop, I was super excited, in part because it existed and also because Uri was actually very inspirational in me getting into physics topics and aging. He was on sabbatical in Boston when I was a graduate student there, gave a course. So I just want to thank you all. And when I asked how I could be involved in the workshop, they asked me to speak about this temporal scaling work, which has now been out for quite a while. But I think one of the reasons they wanted me to speak about it was because it opens up some questions that really haven't been addressed yet. And so there are many fresh parts of it remaining. So let's get into it. So I don't really need to introduce survival curves and lifespan distributions, but I'd just like to emphasize that the sum product of biomedicine over the last, say, 100 years has been to add 18 years onto our lifespan. But 14 of those 18 years are actually under the belt by 1960. So the invention of toilets has done more than all of the molecular therapies that happened in the second half of the 20th century. And why this is, you can read right off of this hazard plot that's been well described before. So you have this period, this is the plot from 1900. You have this period of flat risk. People in their 20s to 50s 100 years ago weren't getting increasingly likely to die. But then after age 50, you get this exponential or polynomial increase in the risk of death. And what we've done over the last 100 years is to drop the risk of death in, say, the typical 30-year-old I show here, 15-fold, which is a dramatic accomplishment. So people are no longer dying of tuberculosis, flu, infectious diseases like they used to. And one of the reasons people see such an opportunity in treating aging is that now 30% of people are dying in this regime of high mortality that's really been resistant to any kind of treatment over the last 100 years. Right? So one of the reasons people are excited about this is for the last, say, 30 years, we've known that you can actually, unlike in humans, you can drop the risk of death in C. elegans by a lot. Right? And so C. elegans is this 1 millimeter long worm, and they live for maybe 3 weeks, but they show a lot of the same mortality dynamics has been discussed in the context of human and bacterial aging. And the idea is, if you have this ability to drop the risk of all-cause mortality so much, perhaps there's some structure to the aging process. So if there are, say, 4 causes of death, that you might see this big complex network, perhaps the fact that there's these single gene interventions that can dramatically change lifespan means that there's some sort of causal structure that we might be able to use for advantage. The idea is if you could find the core determinants of aging, perhaps you could intervene in them and then affect multiple causes of death simultaneously. So this should all be pretty familiar to the audience after many of the talks today. But I think the general idea was put down in the early 90s by 2 scientists who I find very inspirational. And then there's been, along the side, in addition to the experimental evidence from model organisms, there's been a field of biodemography. I think much of what people have been talking about today is the idea is you can treat the lifespan distribution as an endpoint distribution. Right? And the idea is you can learn about the steps going up until the causes of death by how their timing varies between individuals. Right? There's large, very interesting literature here. But the general idea, and I just summarized this for maybe not the physicists in the room, but for the biologists trying to get into this type of thinking, is the idea is if you can have a model of the aging process, it could be a model that produces damage repair. It could be something as simple as the Gavrilova model, where you have the failure of redundant components. The idea is if you can get your model to output a distribution of lifespan, then you can start trying to be empirical in your analysis. And the idea is if you see a Gompertz distribution literature, so in human, worm, fly, bacterial data, you can go backwards and suggest, well, perhaps my model is right because it's predicting the correct lifespan distribution. And I think overall, the fundamental limitation of this type of thinking is the idea that unfortunately, although there's quite a rich phenomenology of shapes of these survival curves, there's not a unique model that would produce a unique lifespan distribution. And that I could articulate, and many have, for example, three separate models of aging that will produce lifespan distributions that are pretty close to what you'd see in your experimental data. Plus or minus some wiggling that might be the result of experimental blurring of your results. And in the case where there's multiple models predicting the same data, you can't really go backwards. And so the idea is, how can you use lifespan data to start supporting these phenomenological models of aging? Right, so fortunately, physics isn't just theory. It's an experimental science inspired, and with this feedback loop people have talked about many times between better theory, better data, better theory being a virtuous cycle. And so there's lots of issues, even in the human literature. Is there a maximum human lifespan? Not. Some of the players have been around today in this. And so just as a philosophical point, like, if we want to have physics make an impact in aging, do we need an Einstein who's sitting with a pen and paper? Do we need a CERN where there's like a data generation? Do we need both? So I think that's something to think about, what type of physics is going to be influential in the aging field. So where I came in is the idea of trying to use these lifespan distributions to inform models, as I said. And the idea is, can we collect better data to perhaps eliminate some of the possible models that are consistent with a better data set, right? So in C. elegans and most model organisms, mice, flies, lifespan data is very precious. And you have a lot of very well educated, very intelligent people sitting in front of the microscope looking at individual worms. Right, so this is a worm that's alive. I think it's a video. I don't know if the video is playing on this screen. I guess, yeah, I have to click on it to play. I guess it's not going to play. Anyway, the idea is worms don't move very much. So this is a pretty good, late in life. So even the video not playing is pretty accurate. But the idea is you can go with a pick and you can poke the animal and then it'll move its head a little bit. And so it actually ends up being very labor intensive to collect the type of data we need to do the modeling. And so spent kind of 8 years-ish trying to develop a new measurement technique, this automated lifespan system that we now have the next generation out a couple of years ago. This is a current graduate student in the lab, and basically you can fill up a hallway with equipment that saves a lot of people a lot of time from doing the measurements automatically. It's basically a big distributed microscope, and the idea is you have these populations of animals. Each one of these is a worm crawling around, and you have some image analysis that's tracking them and then calling their timing of death. So you can then generate these survival curves, as you see in the bottom right-hand corner of this video. And then transition from life to death is automatically identified. And what this does is, I don't know if anybody's ever done 1,000 individual lifespan experiments by hand. I don't know if there's any worm people in the world. Yes. So, yeah. So it's a lot of work. It takes over your life, right, for months. And so we for a typical experiment, this is kind of the data you get. And if you can automate this and scale it up, you start getting a much richer data set. And the idea is that this can better constrain models of aging. Right, so lots of people are using this equipment now. We're very happy that it's been used for purposes other than physical modeling. But when we really got this going, we wanted to start with a quantitative intervention, and we started with changes in body temperature, which are easy to do because you can just crank up the temperature of the incubator, and all the worms get hot, and they age fast and they die young. Right, so what happens to the lifespan distribution under this intervention? And so here I'm plotting the distribution of lifespan. That might be familiar to people who don't look at a lot of cumulative distribution plots. So some worms live short at day 14, some worms live long at day 26. So 20 degrees, which is kind of their natural habitat, you can see this distribution. And as you start cranking up the temperature, you can see this cool kind of thing going on where you're having a transformation that the distributions are moving towards zero as the animals live shorter and shorter, but they're also getting narrower. So there seems to be, the effective temperature seems to be affecting not just the mean, but the variance of the lifespan distribution. And they're even approximately constant within the statistical error you have in these measurements. And so Uri kind of beat me to the punchline here, but there's a very simple way of describing what's going on here, which is if you take two distributions, if anybody's ever used Illustrator, you can stretch objects. You see it makes things thicker and thinner. So if you start stretching the lifespan distribution that we collected at 31, you realize that it kind of overlays very, very well the distribution age at 20. And so when we saw that, like, we're like, oh my gosh, that's really interesting. It's the kind of thing that probably doesn't happen by accident, but we didn't know what was going on. And, you know, we could describe it. The idea is that this type of stretching in respect to time is a temporal scaling. The idea is that there's some underlying survival curve shape, this S, 0, that you have the same functional form, whatever it is, but the way that you map one curve onto the other is just by multiplying each instance of time in that equation with a single scale factor, right? So the nice thing about working in a wet lab is if you can't think about what you're studying, if you don't understand what you're studying, you can just, you know, do more experiments until something comes to you. And so we moved from temperature. We started looking at this insulin signaling receptor mutant in C. elegans. Cynthia had found in '93 that it, you know, can double lifespan. And what we found is when you do the stretching here and doing it with an accelerated failure time regression model, detail's not important, but the idea is when you look at the residuals, you see this stretching. Then we have a gene when you mutate it, the animals are much shorter, HSF1, again conserved in humans. Again, producing a temporal scaling. And changes in diet. So what you can do in C. elegans is they eat the bacteria, but at some point as they get old, the bacteria starts eating them because their gut lining doesn't work really well and they become the prey of the bacteria. So if you UV inactivate the E. coli so the E. coli can't divide, you actually extend lifespan a lot, again, by temporal scaling. Right, and then so we got really desperate, so let's like put these worms in a very aggressive oxidative poison, torpedo peroxide. Again, you get a temporal scaling. So at this point, I was both very excited and also very upset because I had thought that I would be able to distinguish between these different types of interventions based on the transform they have on the lifespan distribution, and said everything was having the same effect. So it was kind of a bit of a cosmic problem for me. But you know, as I became better educated and read more, I realized that, you know, physics is built on the concept of invariance. So data collapse is one way to put it, but then another way to put it is that there are things that, despite the details in a system, properties that remain consistent across lots of complicated things going on. So for example, conservation of energy and momentum are very well understood properties of a physical system that don't change when you know, take two asteroids and smash them into each other, for example. The ideal gas laws, PV equals nRT, tells you how with an ideal gas, if you compress it, it's going to get hotter. All other things being equal. And so these are invariants that we, oh yeah, and also like the speed of light is constant between reference frames. And so you can build very like incredibly cool models of how the universe works built on invariants as their fundamentals, because they let you simplify systems to just the one aspect that's going to remain constant. And this is kind of what you want when you're trying to work on a very complex system like aging. So these invariants we understand. I still don't know why lifespan is being rescaled in C. elegans. But the idea is that perhaps we could find the invariant underlying these lifespan distributions, and that remains an open question and what we're trying to work on. This is published a while back. Right, so this invariance isn't a worm-specific thing. There's like all sorts of pros and cons of any model organism one might be working on. But people have seen temporal scaling yeast bacteria, as mentioned earlier today, and also some of the mice interventions are producing nice temporal scalings, not all of them. So perhaps this is an aspect that's general across model systems. So, you know, when we were trying to write this up and getting feedback, people were saying, well, okay, this is nice, this thing is very general, but can you have some sort of like mechanistic model or phenomenological model that would allow us to understand where temporal scaling might come from? Why is it so general across interventions? Insulin receptor, heat shock factor, changes in diet, and now maybe yeast and bacteria. Like what aspect of these systems are equivalent that would generate the same responses in such diverse contexts? So what we came up with was actually the idea of the network itself generating some of the responses to interventions we're seeing. And the idea is that biological systems, from yeast to worms to humans, they're very interconnected. So we saw earlier some of these beautiful 3D rotating solar system images of protein-protein interaction networks. And the idea is that there are lots of interconnected parts. And some better physicists than me, Maha, and in the Harvard Engineering Department, had just put a paper out showing how just complex networks with lots of engineering components can generate lifespan distributions of shapes. And here's, I think I have a minute left, so maybe I'm just going to go walk you through their model. And so the idea is you have nodes, and these arrows are causal relationships. They're not physical interactions necessarily. And the idea is that the nodes depend on each other. So this blue node depends on these other 3 nodes to function. And the idea is when the majority, and these are binary nodes, are either working or not working, when the majority of a node's dependencies fail, that node fails too. Right? So it's just like, yeah, you depend on your neighbor's function to survive yourself. And so if you start playing Jenga with these networks, removing one node at a time slowly, the consequences of each failure start to propagate, and at some point, you get this collapse. Just like in a game of Jenga, they're pulling blocks out of a tower. At some point, the tower all collapses, right? And you can look at the number of nodes that are functioning over time and then see this collapse, and it's a great relationship, again, between the internal state of the system and the lifespan distributions that I was talking about earlier. And so if you add a little bit of noise into the model, you can get lifespan distributions, and Maha and Viral had shown that these actually generate lifespan distributions that are Gompertzian or exponential, depending on how you tune the parameters, right? So we were excited about this, and we said, what happens if we start modifying parts of the network? And so the idea is you start taking a subset of the nodes and you strengthen them. So you just lower their intrinsic rate of failure, right? So if you just take the whole network and strengthen all of the nodes, you just change the time scale of the whole network, so you get temporal scaling. That's not a big surprise, because you're just changing the one time constant across all the nodes. But what we were surprised to see, weren't really expecting to see, is if you strengthen a subset of the nodes in such a network, the idea is as you increase the fraction of the network that you are strengthening, you also get a temporal scaling. The idea is even you have a node is neighbors to its immediate neighbors, but it's neighbors' neighbors to the entire network. And so strengthening subsets of the network actually has the effect of slowing down the aging of the entire network, just because everything is so interconnected. And so this is the phenomenal model that we have at the moment that would explain why so many different interventions are producing a temporal scaling model, because it's about the network, and perhaps these interventions are all acting on interconnected aspects of physiology, but are serving to slow the decay of the network as a whole, right? And so the idea is to go forward with people who are trying to do mechanistic work in aging is, unfortunately, like the mechanisms people want to study, the causes of aging, are going to be embedded in complex physiologic networks. You can't have a telomere in the absence of a ribosome. That just doesn't work. And so the two are going to be somehow interconnected. And so, unfortunately, this means that we might need to understand the systems that these mechanisms are in before we can really understand the mechanisms themselves, because so much of how they influence the systemic behavior depends on the system and not the mechanism. And so, I don't know, this is maybe a more philosophical point, maybe it's inspiration for why people should be doing more systems work, but I think it's also a warning, which is that, reductionism, in a system that's producing temporal scaling, there's lots of reasons to suspect that reductionist approaches are going to not ever take you from them all the way up to the system, and that you have to understand the system dynamics as well. We've been working on this ever since, so I'm actually speaking again on Thursday if you want to hear some of the works we have trying to map these models down to specific mechanisms. Yeah, so just thank you.
Thank you, Nick.
Yeah.
Got a question right behind you, Brian.
Yeah, I mean, I think that I've sort of come to the same conclusion from a completely different way, which is looking at what these interventions do. And people wrote a lot of papers over the last 10 years saying that whatever your favorite intervention is affects this hallmark. But if you look at it sort of holistically, you find that the interventions, most of them affect most of the hallmarks. And so I think that what it is from a wet lab perspective, we have to get more direct about these interventions. But I think they are preserving the node, but the question then becomes, can we combine them together? And we're not going to combine them together by just saying this one affects this hallmark and this one affects that one, because they're all entrained to each other. And so I think the only way to really try to do that is if you have a drug, to figure out what the direct target of the drug is and get, more defined about the actual activity of the drug, because otherwise we don't know how to combine things together right now, and I don't think looking one hallmark at a time is going to advance that. I think your data is supportive of that too.
Yeah, what is the network? I think you are probably thinking about a molecular network, which you have in cells, but in animals you have hierarchies of networks, right? You have cells that are interacting in an organ, and then you have organs that are interacting within the organism. Do you think these scales matter?
Yeah. So I can tell you what I imagine. So C. elegans has multiple tissue types that all depend on each other. So it's also a hierarchical structure in C. elegans. And some of our work suggests that some of this stuff is actually different organs. I think probably, but we don't know. This model, the idea is these models of relating failure dynamics to lifespan distributions. They are very general, so it doesn't even depend if it's like a random network or a hub and spoke network or something like this. So it's a phenomenological model of how any set of interacting components, under certain connectivities, will generate failure lifespan distributions. So you can project this onto your topic of choice, of your level of resolution of choice. I mean, in my mind, I think of them as cells of tissues, or perhaps as functional components of metabolism or something like this, not individual genes. But
We've got 2 questions here, 3, and then Vadim, and then we finish.
Hi, over here. Yeah. So that was really good to see again and get your perspective on your work again after a while. Really great. So I was just wondering, the only sort of discrepancy we've seen in C. elegans lifespan, but this was like doing meta-analysis, was when we looked at mTOR inhibition, and then C. elegans was the odd one out that actually wouldn't like scale, and the other ones did. So do you know of anyone who's done like a high enough sample size so you could look at that again?
So I don't know about mTOR, but we do have 2, up until this summer, we had 2 examples of interventions that break temporal scaling. They increased variance. One was a mitochondrial mutation, NEO6, and the other one was a starvation approach, E2. And they both increased lifespan variance disproportionately more, and we thought that that was going to be an experimental artifact of a frailty phenomenon. More recently, we actually found that ablation of the germline has the opposite effect, decreasing lifespan variance disproportionately, which is actually quite exciting because it can get you some of the mechanisms that are making some individuals live longer than others. In terms of mTOR, I mean, there's Raptor and Rictor and worms. I haven't seen high resolution lifespan distributions for those 2 mutants. And rapamycin, I think in most experiments, not all of them, it's going to extend lifespan, but it's such a small effect on lifespan, you don't have the statistical support to look for scaling.
Hi. So I'm curious about the other functional readouts as they pertain to health span. So have you looked at whether the scaling behavior also is translated to various health span features?
That's a wonderful question. So we have a paper in PLOS Computational Biology, or it seems like a lot of good quantitative aging research is coming out, where we look at health span. So we built a new version of the software. Worms stop moving at some point, maybe 3/4 of the way through their lifespan. And you can look, that's like an endpoint distribution you can map, and it's actually really interesting. I don't think it's temporally rescaled for our definition of health span for interesting reasons. I think it's a different aging process than the one that's determining lifespan going on within the same individual. But there's a whole paper there. I can send you the link. Colin Ewald, who's here at the conference somewhere, I think he's probably speaking now, found that health span was rescaled, but it was a very different metric. And I'm not really sure how to reconcile those results.
So one of the kind of debates in the longevity field is, if you solve one of these issues, like one of the hallmarks, are you going to see a meaningful extension in lifespan and health span, or do you have to solve all of them to really get any meaningful extension? It seems to me that the model you're describing here implies that strengthening any one subset of the nodes is going to produce some meaningful gain in lifespan. Is that
Absent
correct?
any other complexity, just the network dynamics of things being interconnected will have that effect. So this is like a 0th order naive model of an interconnected system. But of course, biology is often dependent on specific details, not general phenomenon. And so we never know where it is, but like absent any other complexity inside these aging systems, you'd expect temporal scaling as a result of just the fundamental behavior of components in a network.
Last one, Vadim.
So, Nick, very nice talk. So in your study, and of course in many studies here at the workshop, the starting point is mortality. So in a way, age related to mortality. But for some other people, aging may be a loss of function or maybe accumulation of damage or change in biological age. We know that some species have very strange mortality patterns, like if you look at turtles or trees, or like naked mole rat, which doesn't show increased chance of dying with age. How do your models relate to these different patterns of mortality, and in a way, what do you think aging is?
Yeah, so you can get immortal networks very easily by just tuning. So there's a damage component and a repair component in the original Maha model, and if they're balanced, the networks live forever, right? So I don't think that there's a problem or a conflict with the idea of having long-lived or immortal species in this model. You just don't learn as much, because if the experimental data shows that they never age, you don't really have a data set that you can use to model lifespan distributions.
I'm not talking about mortal models. I'm not talking about hydroplanaria. I'm talking about mortal species like plants, turtles, naked mole rat.
No, you can also get flat hazard functions here too. So I can send you a copy of the original Maha paper. They show that there's a variety of different shapes you can generate with the model. I think the expressivity of this network is similar to what you can get from any kind of extreme value distribution, and that you can get arbitrarily flat slopes out of your Gompertz distributions, for example. So I don't think that's like a testable prediction of these models about how flat the lifespan distribution should be, because I think that is just tunable in part by the number of components you put into the model.
Organism age without changes in mortality?
That's a semantic question. So like, what do we want to call aging? You want to call aging the risk of death increasing over time, or do you want to call it something else? Well, I mean, it's a different topic, right? I mean, it's not, we can have multiple different things that are people studying. Some are people studying demographic aging, some people are studying aging of the muscle, right? And it's the same word, but it means different things. Is that your question, or? Okay. Yeah.
Cool, thank you. Thank you, Nick.